So we will now talk about microfilaments, which is the third category of the cytoskeleton. And microfilaments are mainly made up of actin. And actins, as we know, are contractile proteins. We discussed it right at the beginning of the lecture as well. And in presence of myosin, which is the motor protein for actin, we can also have transportation, we can also have movement of organelles and vesicles on microfilaments. But keeping in mind, microfilaments themselves are capable of contraction. So therefore, when we come to cell motility, which is something we are going to be talking about a lot today, in terms of cell motility, the microfilaments become very important. For organelle motility, vesicle motility, microtubules are very important. But for cellular motility, microfilaments are crucial. This is also a very, very energy intensive process in that the polymerization of the actin now uses ATP. So remember, for microtubules, we were using GTP. By the way, I forgot to mention that microfilaments, when they are assembling, remember the assembly of the microfilaments, intermediate filaments that we saw in this picture. This particular process of assembly of intermediate filaments does not require any energy. So this does not actually involve any energy usage for the cell. But for the assembly of the microfilaments, which are made up of actin, there is a need for ATP. So the globular actin or G-actin polymerizes to form F-actin, which is the filamentous actin, and it is carried out with the help of the breakdown of ATP for every single globular actin that goes and binds to the polymer. Again, there is directionality. The two ends of the actin filament have different structures. So there is a plus end and a minus end, which is also called a barbed end and a pointed end. This barbed and pointed characterization is based on the binding of, this is an experimental observation whereby the actin filament looks like a barbed end or looks like a pointed end. And what we mean by a barbed and pointed end is, if you think of an arrow, so I'm trying to draw an arrow here. So this is the barbed end and this is the pointed end. So it's really difficult to imagine how the actin filament would look like this. But apparently when people observed actin filaments using labeled, fluorescently labeled myosin fragments, then the actin filaments looked like they have a pointed end and they have a barbed end. I personally prefer using the plus end and the minus end because it makes more sense in my head. The plus end is where more of the globular actin monomers can be added, and the minus end is where the actin monomers can be removed. So the two ends again have different structural and dynamic properties, and so there is a direction on the actin filament and movement can happen on it. So here is how the actin filament looks. So this particular actin filament is made up of a number of globular actins, and you can see here there are two actin filaments here. Okay, so this is the first one and this is the second one, and the two actin filaments have formed a helical structure here. Okay, and this again gives quite a lot of, and you can see the helical structure over here too. So this is one of the actin filaments, this is the second actin filament, and they have sort of wrapped around each other to form this nice helical structure. And this is how a filament looks. Microfilament assembly and disassembly has again been identified in vitro. So again, whether the exact same mechanism occurs in vivo or not is something that will need to be examined maybe in the future if the microscopy technology improves dramatically. But actin assembly and disassembly in vitro depends on how much actin monomer is available. This makes perfect sense because if there isn't enough actin monomer available, obviously the assembly will become a little bit delayed, right? Because there isn't enough monomer. Filament assembly leads to a drop in ATP actin. So what this means is in a cell, let's say that I'm drawing a cell over here. So in a cell, let's say this is a cell, we have some actin filaments being formed. So I'm just going to draw them as lines. And then we have a lot of globular actin. So these globular actin are found in depose inside the cell. And these globular actins are actually, if I draw a globular actin like this, they are ATP bound. Okay, so when they are ATP bound, they can go and bind to the filamentous actin. The ATP will be degraded to ADP and PI, and then the globular actin will be added to the filamentous actin. So filament assembly, therefore, will lead to a drop in the amount of free ATP-bound globular actin or monomeric actin that may be found. Actin subunits are added to the plus end, and then they are removed from the minus end. So we will see some pictures showing us a steady state activity whereby continuously plus-ended addition and minus-ended removal leads to, this is called treadmilling, leads to a steady state in the length of the actin subunit. The microfilament or the actin cytoskeleton is organized by controlling the equilibrium between assembly and disassembly of the microfilaments. There is nothing like the catastrophe that we have seen for microtubules. Rather, there is a regulation on how quickly or how much more assembly there is compared to disassembly. If there is more assembly, obviously the length of the filament will increase. If there is more disassembly, the length of the filament will decrease. So when we look at this picture, this is a very, very nice picture. This is completely in vitro. Okay, so whether this actually occurs in vivo or not, we don't know. If you take a cell-free system, so at this point I would like you to understand what we mean by a cell-free system. This is where the membrane has been discarded, globular actin has been isolated, and then if you use a short actin filament, that can act as a seed. Okay, so this is the template similar to what the gamma tubulin was doing for the alpha beta tubulin. This is the seed or the nucleation on which the new filaments can be, new globular actin can be added. So again, here we have a minus end and we have a plus end. And this is very, very structural. The shape is such that it allows the addition of globular actin rather than any specific chemical change. This is a structural change that we have here in the plus end. So we generally, if you add a very, very high amount of ATP actin, so this is the globular actin that is ATP bound, in general, when there is a very high amount of globular actin, remember this is again happening in vitro, we can have addition at both ends, plus end and minus end. But the addition at the plus end will be much, much higher. Okay? So this is when we have a high amount of globular actin available in this experiment. As more and more globular actin get added, and remember, for it to form the filament, the globular actin has to hydrolyze the ATP, and now it has become an ADP-bound ADP-bound actin filament. Now, as the concentration drops, we will continue to have addition on the plus end, but the addition at the minus end will then become a little bit weaker. As the concentration drops even further, we will again have even less addition at the minus end. In fact, now we will have removal at the minus end because there is very, very little of the monomeric actin available in the cell. Here, the color is not from ATP or ADP. The color is from monomeric actin or filamentous actin, or in other words, the new actin that has been added and the actin that has been removed. So here then, as the concentration drops even further of the globular actin, the monomeric actin available, we will have complete loss of addition from the minus end. We will only have removal from the minus end, and we will have addition at the plus end. So as we add on the plus end, we remove at the minus end, and now the length of this particular actin filament remains constant. This is called treadmilling, where, as you know, you can walk on the treadmill in the gym. The treadmill doesn't get longer, but you keep walking for many, many long distances. You can walk for kilometers at a time, right? So, but the length of the treadmill doesn't change. So here, this is also called treadmilling, whereby you have now a steady state whereby the number of globular actins that are being added at the plus end is equal to the number of globular actins that are being removed at the minus end. When the concentration increases again there will be more addition at the plus end and less removal at the minus end. Oops. So so we have the treadmilling here. Um, when we discuss about the proteins, the motor proteins of actin, myosin and actin interaction is very well known. It's like a match made in heaven. So just like streptavidin and biotin can interact very, very nicely, or an antigen and an antibody interact very nicely, myosin and actin is one of those couples that interact very, very well. So myosin is the molecular motor for actin filaments. Myosins are of many categories. There are the conventional myosins that you have learned perhaps in your high school or even in your first year of how the muscle contraction happens. So those are the type 2 or conventional myosins. We're not going to be talking about those myosins today because you will learn about it from me next semester in your physiology lecture. So the myosin that is involved in muscle contraction is not the myosin that is necessary, that works, that functions in the cell as a molecular motor. It is the unconventional myosins, typically the type 5 myosins, that function in cells for the movement of vesicles or organelles. So all myosins, no matter which category they belong to, they have a characteristic motor head, just like we saw for dyneins and kinesins. These also have a motor head that can bind to their target, in this case it's actin, and they can also hydrolyze ATP. So remember, just as kinesins and dyneins required ATP, myosins also require ATP in order to move. They are motor proteins. Motor proteins, by definition, require some form of energy. The tail is what is different between all the different groups of myosins. So the conventional and unconventional myosins can be found in different cell types as well. So conventional myosins are the ones that generate force in muscles, and there are some non-muscle cells that also can have conventional myosin. Conventional myosins have two heavy chains, two light chains, and two globular heads, which are the catalytic sites. So as I mentioned, we are not going to be talking much about myosin 2, but just as a picture, this is the tail, and we have the head here. The head has the ATP binding domain and the ATP hydrolysis domain. And we have the light chain over here, and there is a neck. So this is how the heads look like. This is the structure of the myosin. And the long tail is what will form the thick filaments that you perhaps remember from your previous lectures as the thick filaments in a sarcomere. Uh, the motor activity is located in the head, and this head, there are several experiments that have been carried out with the head, which is also called the S1 fragment. In fact, this S1 fragment, if it is conjugated to a reporter, such as a fluorescent protein, then one can actually visualize the function of actin very nicely, because the S1 fragment will go and bind to actin with high affinity. So the motor activity is contained in the head, and the tail portion, it basically has a structural function which allows the formation to form the filament, which we know as the thick filament in the sarcomere of the muscle. So when we look at the movement, this is a very, very nice picture. Again, we have the head, the S1 fragment that we saw a couple of slides back. So this is the S1 fragment. If you were to immobilize the S1 fragment on a glass slide, and this is a real experiment that was carried out. So the S1 fragments have been immobilized on a glass surface and then washed. So any non-immobilized myosins are removed. And then the glass slide is incubated with the fluorescently labeled actin filament. So you can see the actin filament here. The helical structure is also very nicely visible. And you can actually monitor over time with using time-lapse microscopy. You can monitor whether this particular actin filament has moved or not. And you can see that this particular, from the left side, you can see that the one of the actin filament has moved quite far. So each and every one of them have moved, suggesting that it is the myosin S1 head that is responsible for the movement of actin. And this is something that is happening all the time in our muscle cells as we contract our muscles. So here is how the heavy chains are, and this is what is forming the thick filament of the myosin in the sarcomere. But now we will come to the unconventional myosins. The unconventional myosins are more relevant to our discussion today. They have a single head and they cannot form filaments. So this filament structure, that's why I kept this slide, this filament structure that we see in type 2 myosins, which are seen very, very commonly in the sarcomere, this filament structure is missing in the unconventional myosins because there is absolutely no need to form the filaments. The myosins here are there, here are functional simply to carry vesicles or organelles. Myosin 5, as I was mentioning, is primarily involved in organelle transport, and many of them are associated with vesicles and organelles. So here you may ask me the question, and I'm sure one of you would have come up with this question, is how do we decide which particular, what do we use? Do we use microtubules? Do we use microfilaments? How does the cell decide? So one thing that has been shown very nicely is, let's say that this is, I'm sorry, I should be drawing here. So this is the axon. A lot of these experiments, as you can imagine, have been done with axons. So we have the microtubules over here. This is something that has been shown over and over again. But when we come to the edge of the axon terminus, they become microfilaments. Okay, so here we have a lot more microfilaments. So it seems that the vesicles that have been brought up to here with the microtubules are then now given to the microfilaments, and then the microfilaments will take, or the motor proteins on the microfilaments are the ones that will take these vesicles right until the edge of the membrane. So there is some handover of vesicles as well as of organelles, if necessary, to from microtubules to microfilaments at the edge of the cell. This has been shown in experiments. Okay, so far I hope we are okay with the idea of microtubules and microfilaments, and I'm going to take a very, very short break here to show you a very interesting video before we move on to non-muscle motility. So very quickly, I'm going to talk about non-muscle motility. There are a few slides here, but before we do that, I want us to watch a quick video just to give you a quick break from this lecture. Microtubules and microfilaments are connected. No, they are connected. So remember the plectins? So we talked about the plectins earlier. So there are proteins which can connect the microtubules and microfilaments. Are you able to see the screen now? Please somebody turn on your microphone.
Yes, hocam, screen is visible.
Okay. So in non-muscle motility, we are talking about actin-binding proteins, which affect the localized assembly or disassembly of the actin filaments. In the video that we saw, you did see one example of a severing protein, which goes and cuts the actin. We will talk about several of these actin-binding proteins now. Let me see what this page is. Okay. All right. So there are many different types of actin binding proteins. And if we start from number one here, this one, monomer nucleating protein, is a protein that helps in the nucleation of the actin. Remember in the in vitro assay, the cell-free assay that I described before the break, we saw that there was a small strand of an actin filament which acted as the nucleation factor there. But in the cell, there are proteins that are called monomer nucleating proteins where the actin filament will start getting synthesized. And as an example, I can give you the example of a protein called ARP23. Okay, so this is a monomer nucleating complex. There are other proteins called monomer sequestering proteins. So this that's shown in little green here, this is a monomer sequestering protein, which means that it will sequester the monomers, it will keep the monomers combined with it, it will complex with the monomer, and it will prevent this monomer from forming an actin filament. So this is again a very close, very sophisticated level of regulation whereby the actin filament will not be allowed to elongate in that particular region. As an example, it's thymosin. This is another protein which acts as a monomer sequestering protein. Cap proteins, which you can see over here, these are very, very common. This basically keeps this actin filament in a certain length. Okay, so this actin filament will not have any more monomers added to it, so that the length of this actin filament is kept constant. These actin binding cap binding or capping proteins are seen very, very commonly in the sarcomere in our muscles. And as one example, I can give you CAPZ. This particular protein is a capping protein that is found on the Z discs of the sarcomere. Just as a quick reminder, remember we have the Z discs. So we have these thin filaments which look like this, and then we have some more on the other side. This is a horrible, horrible diagram. Sorry about that. And then we have the thick filaments over here. This is how the sarcomere looks, right? So we can have the capping proteins at the Z disc edge as well as at the middle of it in order to prevent the length of this actin filament from getting any longer. Monomer polymerizing protein. So where do we have? Yeah. So we have the monomer polymerizing proteins, which means that these monomers tend to have the ADP replaced with ATP. Okay, so if you have the ADP on the monomer replaced with ATP, then this monomer can now enter into the pool of actin monomers that can be added to the polymer again. So again, there is an example for this protein, which is profilin. So profilin is a protein that will exchange the ADP for ATP on the monomer so that that ATP bound monomer can go and bind to a polymerizing actin filament. So this will enhance actin filament elongation. Then we have a depolymerizing protein. So these depolymerizing protein will enhance depolymerization generally at the minus end. So these proteins will shorten the actin filament. And as an example, we have cofilin. Again, you don't need to remember all these examples, but just to give a name to a protein, I'm giving you these names because they do exist in our cells. So cofilin tend also to fragment the polymers of actin, so they tend to cleave the actin, but they also tend to enhance the depolymerization at the minus end. Then we have these cross-linking proteins that you can see at the top here. You did see the structure very nicely in the video that I shared with you. So these cross-linking proteins can keep again the structural support of the actin filaments, and one of the examples is a protein called vilin that is found in microfilaments, that are found in microvilli of intestinal epithelial cells. Probably we have a picture of it. If not, I'll draw it in the next slide. And then we have filament severing proteins, which is found in number cells. So these are the ones that we saw in the video. An example of this is gelsolin. So this sort of protein will go and cleave the microfilament, so that the microfilament is now shorter. And then we also have membrane binding proteins. So a lot of the cytoskeletal elements are found just underneath the membrane and the cytosolic phase. So the membrane binding protein tends to bind the actin at the focal adhesion complexes. We'll learn about focal adhesion complexes in a couple of weeks, probably at the end of December, where a lot of signaling, outside-in signaling happens. So remember, you saw some integrins in the video. Those integrins would be located somewhere here. So the integrins will be binding to collagen that is found in the extracellular matrix. It will signal through the focal adhesion to the actin complex, giving signals from the outside to the in. There are a number of examples of these sort of proteins, such as vinculin. A lot of these are actually oncogenic proteins, in fact. Vinculin or esrin, there are some very interesting names. These are examples of proteins that help the actin filament to bind to the membrane. Okay, we have a question. Green circle in number. Yeah, so I just described that. So that's the vinculin or the esrin. Okay, so now let's move on. And we will So yeah, so there are these examples are given here as well, nucleating proteins and blocking proteins. So let's skip through this. And one of the examples that I gave you was of vilin. This is a filament, cross-linking protein. And these are found in the microvilli. So microvilli, if you recall, are found in differentiated epithelial cells. So if we look at an epithelial cell that we have in our gut, I keep drawing it like this. So these are the microvilli, extensions of the membrane, which increases the surface area. So inside these membranes, so if I draw a microvillus like this, inside these membranous extensions, we have a lot of actin filaments, which gives them the stability and the shape. And these actin filaments are actually cross-linked with vilin, and this makes these actin filaments very, very, very stable, and they are very long-lived until the cell actually dies. And this extension will allow the expression of membrane-bound enzymes, as well as for the absorption of nutrients into the cell. The end cap here is again stabilizing the actin filament, and then these cross-linking proteins that you see here will be stabilizing the microfilaments themselves. So here is another very clear difference between cilia and microvilli. So cilia, remember, were formed of microtubules, whereas the microvilli are composed of actin filaments. So when we come to non-muscle motility, uh, non-muscle motility and contractility generally comes from actin polymerization as a force-generating mechanism. So as I keep mentioning over and over again, actin itself can have some amount of contractility. So although myosin has much more contractility than actin, but actin itself can also generate some force. So here is a beautiful, beautiful, remarkable example of a bacterium called Listeria. Listeria monocytogenes, or Listeria, for if we call it just Listeria, is a bacterium that can enter inside cells. This is a distinction that I would like you to keep in mind if you don't already know it. Not many bacteria can enter inside cells. Bacteria are generally able to release toxins outside the cell, which will activate inflammation in our body. To enter inside the cell, the bacteria really needs to be quite unique. Bacteria can inject their toxins inside the cell. They can inject their toxins outside the cell, which can affect it, but entry into the cell is quite rare. So these bacteria, Listeria, they can enter inside the cell. They can in fact enter inside macrophages, the cells that are there to kill them. So these are really dangerous infections. And when they enter inside the cell, they actually encourage actin monomerization in their tail. So you can see here beautifully the actin monomers that are found in the cytoplasm of the macrophage. And remember, macrophages are highly motile cells. So clearly they are able to, they do have quite a lot of actin monomer available. And these actin monomers are then taken, and with the help of this nucleating protein, remember we talked about ARP23, that protein is recruited by this bacterium from this eukaryotic cell, and then it encourages this polymerization of the actin, which then moves the bacterium inside the cell. So this is another, it's so remarkable. Inside the cell, it's very crowded, right? So you all appreciate now that movement inside the cell, unless you have energy, is quite difficult. But this bacterium needs to move inside the cell. So what it does is it recruits actin, and then this actin will propel the bacterium further inside the cell. And you can actually see that real picture here. In red, you have the bacterium. In green, you have its actin tail, which it has recruited from the actin monomers that are found inside the host cell. So it's an absolutely remarkable piece of engineering from this Listeria. So cell locomotion is also something that is largely carried out by actin filaments. So this is a cell that is moving, for example, a white blood cell or a fibroblast that is capable of movement. It tends to have a leading edge. So the leading edge is right towards the front. And this leading edge tends to be quite ruffled. So this is also called a ruffled edge sometimes. And then there is a tail which is sort of dragging behind the moving cell. So the leading edge is over here, and the lagging edge is called the tail. So it crawls over a substrate, and this is how it looks. There are specific foci at which it can at which it can attach to the substratum. And when this leading edge moves forward, it generates these interactions, and then the tail of it sort of follows behind. So now we have the original shape of the cell again, and then again it's going to it's going to extend a pseudopodium. This is called a false leg, a pseudopodium, and then that extension will cause the cell to move forward, and then the lagging edge will basically follow it. So this will be repeated over and over and over again until the cell reaches its final destination. This movement is far, far slower than what we learned, for example, for the motor proteins. The motor proteins move very, very, very fast compared to actual cells. So cells that crawl over a substratum, obviously we do need substratum because there has to be actual interaction and signaling between the substrate and the cell. So a lot of these interactions are focal adhesion junctions that we will learn in a few weeks' time. And so there is actual signaling going on, communication going on between the protrusion, which is also called a lamellipodium, and force generation in the lamellipodium occurs by actin monomers. So here, this edge that is being formed here, how is this edge pushed forward? Is this pushing forward happens because of the polymerization of actin filaments. So actin monomers are added to the filaments, and it provides anchorage to the cell, and it also pushes that edge of the cell forward. So here we have yet another picture, very beautiful picture. This fan-like structure is the leading edge, where we have huge amounts of actin filaments being formed, and then the lagging edge. So again, you can see over here, very nice ruffled edges where a lot of actin polymerization is happening. This is the last figure that I will show. So here is a rather nice experiment. This picture on the right here is the edge of a pipette tip. And this pipette tip is being used to inject a growth factor. And you can see this white blood cell here is moving towards it. So this is a signal that is attracting this white blood cell towards that pipette tip. And you can see here, this edge is panning out, and you have this lagging tail that has been formed. So what is happening most likely in this case is there is a signaling that happens. Obviously, the cell needs to understand it. And this signaling is carried out with the help of a protein called WASP. And the WASP, it helps. So this little circle here is the WASP protein, which then recruits this ARP23 complex. The ARP23 complex, remember, is the nucleating complex for the actin, and it results in the addition of actin to the end. So at some point, we will have to stop any more addition so that it has to be regulated, right? So you don't want one part of the membrane poking and then nothing else happens to it. So here we have again the barbed end. This is the plus end where addition can happen. You can have nucleation happening over here. So in the next stage here, we have a capping protein, which will prevent any further addition of actin monomers on this particular, but then here we have yet another one, another actin filament that is being formed. Now this is pushing this edge forward, as you can see here. And again, we have pushing of this edge forward. So we have another nucleation point here, etc. So in this way, we can have extension of the membrane in the direction in which the cell needs to move. And obviously, these actin binding proteins are there in order to orchestrate and in a very, very regulated manner exactly where extension will happen and exactly where disassembly of the actin will happen and where the actin actually has to remain stable. Okay, so I am going to again stop sharing, stop recording.